Today I'm going to talk about something a little different than I normally do, and not actually present any results per se. Instead, I want to try and stimulate you to think about some things in a slightly different way, specifically around what engineering and evolution are and how they might fit together, how current biological design approaches often lack open-endedness that you see in, say, natural systems, and finally show you how important evolution is for the full engineering life cycle, especially when engineering biology is actually deployed into the real world and made use of. Now, it's really important to stress that the work I'm about to present has really emerged from a long-running collaboration between one of my soon-to-graduate PhD students, Sim Castle, and Michiel Stoch, a newly appointed assistant professor at the University of Ghent in Belgium. And this has really been one of the most fun, I think, research areas I've been able to work on so far in my career. But before I start talking about some of the evolutionary focused work, I wanted to first give you a bit of background on my own kind of research interests. So my research is predominantly focused in the area of synthetic biology, and it stems really from a fascination in the ability for a sack of chemicals like the one I've shown here to be able to sense its surroundings, process that information, and then respond by altering its biochemical makeup and broader behaviour in kind of an appropriate way. Now, I originally trained as a computer scientist, and so when I learnt about this area of biology and how it works, it was just really too appealing not to try and develop my own biochemical computations using living cells. Now, a challenge you face when trying to actually do this is that the multi-level complexity of a cell makes kind of knowing where to even begin really difficult. And so to get started, my group has attempted to ignore much of this complexity, and instead we focus on creating simple low-level biological circuits that can control how genetic information, which is encoded in a cell's DNA, is actually physically read. Now, to do this, we can make use of some basic genetic parts like promoters and terminators. So promoters enable RNA polymerase, I've shown by these little moving red blobs, to bind and start reading DNA, while terminators cause them to halt and to disassociate. And so by positioning promoters and terminators in different parts of the DNA, we can actually affect where RNA polymerase flows are able to travel, and therefore how the DNA itself is actually being read. Now, if the DNA encodes for a regulatory element, such as a transcription factor, that then affects the strength of another promoter, we can actually connect the flow of RNA polymerase in one region of the DNA to the flow in another and actually implement basic genetic devices that can carry out computational operations. Now, in this case, this corresponds to the implementation of a simple, what's called a NOT gate, or basically an inversion of the input signal. But the key point here is that by using the flow of RNA polymerase as a common signal in these circuits for both the inputs and the outputs of the devices, we can then easily connect many genetic devices together by merely making the output promoter of one device the input promoter of the next. And pretty much by using this approach, this is how more complex regulatory networks like this repressolator circuit have been produced. Now, this circuit is able to generate more complex functionality, so in this case robust oscillations in gene expression. And I guess over the past kind of over 2 decades now, we've actually become pretty good at building small circuits like this composed of, say, a handful of regulatory elements and links. But one of the surprising things is that we still struggle to create large and robust genetic systems, which is really going to be crucial if we ever want to push our technologies towards engineering, for example, entire genomes of an organism. Now, the reason for this difficulty is that as the complexity of our circuits grows, so too do the number of ways that it can break. And this can include things like cryptic promoters being accidentally included into part sequences, we just didn't realize they're there, parts might just fail to work completely. That happens commonly in a lot of biological systems. There are contextual effects that cause parts to actually alter their performance, even though the sequences might be identical. And there are often uncharacterized interactions with the host cell itself that's actually executing these genetic programs that we're developing. Now, in all of these cases, figuring out how a circuit that's composed of maybe tens or even hundreds of parts has actually broken by just observing how it's behaving is pretty much impossible. Now, when you look at other engineering fields, they also face these sorts of challenges, but what they've typically done is to develop tools like debuggers or multimeters to help with this task. But as bioengineers, we often lack an ability to actually see the transcriptional flows within our circuits, and therefore we really find it difficult to precisely understand why they're actually breaking. And so my research group has really aimed to tackle this challenge in two main ways. So the first has been to develop what we call genetic debuggers and other tools that allow us to have more advanced cellular analytics. And the idea here is to provide a more holistic picture of what's actually happening inside an engineered cell, and using that information to build new types of predictive model to actually aid in the design process itself. Now, unfortunately, the complexity of biology often gets the better of us, even when we can observe what's actually happening. And so also over the last kind of 3 or 4 years, we've also been thinking about how perhaps we might need to evaluate our design approach completely, and whether there might be ways of actually harnessing evolution itself, which clearly can develop incredibly complex biological systems on its own. And I think that really nicely now ties me into the first part of this talk, where what I want to do is to hopefully get you thinking about what engineering is and whether biological engineering is special in some way. So when we think about what engineering is, what we typically arrive at is a cyclic process that involves some form of design step, where a system is imagined to fulfill a requirement that we might have. And in traditional engineering, this might be informed by things like brainstorming, or we might use simulation of models and some optimization techniques to come to a final design. Then once we have a design that we think might work, we can then build it and then finally test it to see if it meets the actual specification. Now, in engineering fields where components that are used are pretty well understood, this one of these cycles may be enough, but almost always there is a gap between the real world and how the system behaves physically and the mathematical models that we use to predict its behavior, which typically means that designs often need to go through further engineering cycles that learn from the testing that's previously been performed. Now, biological engineering has also been adapted to this classical engineering cycle, but because of the larger reality gap and the more unknowns, it's common that many more designs are actually taken through this process concurrently in order to find a working system. Now, interestingly, there are a lot of similarities between this cyclic process and evolution. So, for example, genetic variation in evolution is used to explore new areas of design space. Populations of new designs are then built through development and kind of the embedded genotype to phenotype maps that they encode. And then selection kind of acts as a test to the feasibility of each design, only allowing those that meet some minimal criteria the ability to progress to the next cycle. And something we've been considering is whether we can actually harness this inherent capability for all living things to be able to evolve, working with this kind of special feature that biology has, which we don't see in other engineering substrates. So in this context, rather than, say, engineering a solution to fulfill a requirement in this kind of classical way, we instead want to be what we've called meta-engineers and actually engineer the evolutionary design processes themselves, like, for example, in directed evolution. And what this perspective does is it helps resolve, we think, a tension that is sometimes seen between classical and evolutionary-based engineering approaches, and helps to see them both part of a similar self-refining cyclic process for a system with different inherent capabilities. Now, to try and unify this idea a little further, we've been thinking about how different engineering and evolutionary design methodologies fit together, and specifically in terms of like two core characteristics that they all share. Now, the first of these is what we call the design throughput, which is just the population size that can be effectively worked with during each iteration of this cycle. And the second is the design time, which is the number of design cycles needed for a suitable working design to typically be found. So this diagram that I'm showing here brings these elements together and shows what we've called an evolutionary design spectrum with some of the key methods for design overlaid on top of it. Now, this is very much a work in progress, so any input that you have or ideas would be really, really welcome. But I think what at this stage it already starts to illustrate is how increasing knowledge can open up more powerful design approaches. This is going from the top right to the bottom left. It also shows how traditional design and evolutionary approaches are spread across this space, and perhaps what I think more interestingly is where there are actually gaps. And we've been wondering if these gaps might be right for the development of new design approaches. So, for example, robotic automation could potentially fill out this bottom region near the x-axis, enabling much quicker and numerous design cycles, but typically with a lower throughput than you would see with, say, directed evolutionary type studies. So that was the first part that I wanted to sort of get you thinking about engineering and evolution and how they're perhaps connected. For the second part of this talk, I wanted to highlight the diminishing returns we typically see in the performance improvements of designs when carrying out classical engineering or using approaches like directed evolution. And in particular, highlight the lack of open-endedness many of our current engineering design workflows actually display. So what is open-endedness? Well, an open-ended process is one that is said to have no end, and one that typically leads to continual improvements, increases in complexity, and new innovations. In a biological context, kind of the most well-known process that is thought to be open-ended is obviously evolution itself, where you see this continual increase in the diversity of life over time. Now, there are a number of hallmarks that often open-ended processes exhibit, and these include things like the ability to develop new types of entity that have novel functionalities or interactions, and an inherent evolvability where the system itself actually evolves to make better use of evolution, the display of major transitions like the emergence of, say, multicellularity from single-celled organisms, and finally semantic evolution, where the function of the entities in a system can actually be repurposed in very different contexts, and therefore reused in new ways to kind of diversify the types of things that a system can do. But I guess the question is, how does this fit into classical approaches to biological design or directed evolution? Well, in those contexts, the design or evolutionary process can be viewed as a type of search over the kind of astronomically large space of all possible biological designs. Now, in something like directed evolution, there's always a clear objective or goal, with improvements in performance of a design in a population used to actually guide the designs that make it through to future cycles. And what this leads to is a search process that climbs towards local regions of design space with higher functional activity. But what that also does is typically lead to drops in the diversity of the designs that we see, because you're reaching this kind of single design with peak activity as you kind of move through this space. And what this means is that there might well be vastly different types of design in the design space, but they're not found by this process. Now, an alternative approach that's been put forward by researchers in kind of AI and artificial life, and is really, I think, nicely described in this book by Stanley and Lehman called Why Greatness Cannot Be Planned. And the idea they put forward is that in order to support more open-ended search, it's crucial that there's actually no objective in the search process itself. And instead, the search should merely follow directions that appear novel and interesting at that particular time. Now, this might sound like a really fluffy concept, but actually you can formalize this idea mathematically, allowing you to characterize the novelty of designs and how that novelty changes over time. And this can then be used to develop search processes that explore not only high performance designs through kind of hill climbing processes, but also those that achieve similar functions in very, very different ways. Now, an open-ended search process is therefore powerful not because it can quickly optimize a design by, say, tweaking small features, but because it helps to uncover much more important, what's known as stepping stones that lead to completely new types of solution that then themselves can also act as kind of the basis for new discoveries and new stepping stones. And a couple of examples that I like to use to help explain this concept relate to kind of our search over the history of biotechnology for kind of precise genome editing capabilities and abilities to easily regulate genes within a cell. Now, in both these cases, recently developed solutions like prime editing and CRISPR interference and activation both lean on earlier discoveries of CRISPR-Cas systems. So CRISPR-Cas in a way acts like a stepping stone to these new innovations and approaches. But an important thing to recognize is that if an objective search had been performed before the discovery of CRISPR-Cas, it's very likely that neither of these approaches, or potentially any good solution at all, would have actually been found. And this stems from the key point that Stanley and Lehman put forward in their book, which is basically that if you have really ambitious objectives, which you often do in biological design, these are typically what's known as highly deceptive. And so aiming for them through an objective can actually be harmful to actually finding a suitable solution. And so in these cases, merely searching for interesting or novel features of the design space may actually be more fruitful in getting you closer to a point where you have the necessary technologies to make that simpler step towards the actual solution that you actually need. So how can we ensure that some of our evolutionary design processes maintain this open-ended quality that you see in nature? Well, it's obviously thought that natural open-endedness can stem in three core ways, or not three core ways. It's typically stems from having three core elements, it's thought, within the system itself. And these include things like genetic variation, selection and phenotype realization, plus interactions between these elements and the broader environment. So this already gives us inspiration for new features that can potentially be incorporated into our design methods. But synthetic biology also offers some really interesting tools and capabilities to actually further enhance or modify these elements. And so one of the things that we're starting to do is actually map out and carrying out some initial experiments to see how far this perspective can take us. But it's already, I think, refreshing to see how synthetic biology and evolution can be fused through this idea of enabling open-endedness in the design processes that we're actually building. Okay, so to end, I wanted now to kind of shift a little bit and look more broadly at not just the design, the initial design part of the process, but actually now the entire engineering life cycle, and in particular the kind of the unavoidable but typically overlooked role that evolution actually plays when we deploy our biological systems into the real world. Now, to try and show you the kind of challenge that I'm referring to here, what I've drawn is a schematic of how biological design is typically performed, so related to kind of what I spoke about earlier. And I'm going to call this biodesign version 1. And so you normally would start with a number of candidate designs, and you explore some of these further by maybe tweaking various aspects. Some of them might be dead ends, others might spark connections, and ultimately they'll lead to a final design that displays the behavior that we want. Now, this exploration phase could be performed manually by humans, but more recently it's also been a shift to carrying out this kind of design search using what's known as biodesign automation tools. But ultimately it's this search phase that when we talk about design, that's what we typically mean. Now, in most engineering fields, a design won't change when used, but for biological systems this isn't the case. A biological design can evolve over time and actually deviate from its original function. And so there's a period of what I'll call artificial evolution, where we come up with a design, followed by a period of natural evolution, where the biological system is actually put to use. And in addition, there's also normally an operating window over which a system is said to be functional. And so if evolution doesn't push the system outside of this region, we don't need to worry. But as the functions that we typically implement in cells often impact the ability for the host to grow, virtually all the systems that we build will eventually break, and we just basically hope that they don't break before their kind of desired lifespan is actually reached. Now, what my lab has been thinking about more recently is how a biological design workflow might look if we took a more complete view that integrated both the artificial evolution of our own kind of engineering design process and the natural evolution of the biology once it's been deployed. And this sort of approach would allow us to think up front about the size of an operating window and the ways that we might be able to reduce the ability for evolution to actually affect our design over the desired lifetime of the system. Or I think way more interestingly, it also might allow us to think about how evolution itself could be harnessed to automatically search out new functionalities when needed due to, say, potential shifts in an environment. And it's this type of adaptation that really touches on one of the unique characteristics of biology that I kind of highlighted at the start, but we haven't really yet figured out how best to engineer with it. But I really do think it's going to be crucial if we want to harness kind of biology's full potential in many ways. Now, to be able to effectively describe and reason about biological designs in this way, we need a conceptual framework, and this is what Sim Castle has actually been developing as part of his PhD, and he calls this framework basically the evotype. And the idea is that the evotype is a way of capturing the evolutionary potential or capacity that a biological design has. So to understand the evotype, we lent heavily on the idea of an adaptive landscape, but altered it in a number of key ways. So we start with this 2D landscape that I show here that is used to represent the sequence space around a design, so it's a little red dot. And then we use 3 core aspects that shape the artificial and natural evolution of the design. So namely, the possible variation that can occur within the system, how well the design's function actually meets our desired goals, so basically its utility, and then finally the natural selective pressures that are present affecting its reproductive success that ultimately underpin selection. Now, to combine this information, we first consider the variation that's actually possible. Now, this is made up of many different processes that can lead to mutations or changes in the genotype of the biological system that we're designing. And some of these introduce small changes, like, for example, slip strand mispairing, while others might result in really large modifications, which are typically called algorithmic jumps in sequence space, like those that you might see during recombination of, say, repetitive sequences. Now, for an evolving biosystem, each of these processes combine together to generate what we term a variation probability distribution that spans over the sequence space. And so this provides a probability that the design type will jump to another part of sequence space during evolution, and we represent this probability by the darkness of the shading across our evotype landscape. So we're now able to kind of represent the possible routes that evolution might take, but evolution obviously isn't just shaped by variation. Both the artificial and natural selection I mentioned will also play a role. And so, unlike in an adaptive landscape where the height is typically related to the fitness of a sequence, here the height corresponds to something that we've called fitneity. Now, the idea behind fitneity is that it captures a combination of both the artificial and the natural evolution present within a biosystem, which is really governed by what we call the utility and the fitness that drives them. And it can be sort of as a way of calculating how good a design is that trades off both of these factors. So, for example, every design we create needs to perform some function, and how well it performs that function is basically its utility. So a high utility is obviously good. Now, similarly, because this is a living system, a good design also needs to allow the organism to reproduce and remain viable, and so a design can't cause excessive stress. So a high fitness is also seen as good. But as it's really rare that both of these aspects can be maximized simultaneously, and typically they actually oppose each other in many ways, fitneity captures how much deviation in utility and peak fitness we're willing to accommodate, basically how well that particular design meets our own and its own needs. So ultimately what a bioengineer is trying to do when you view it in this way is not only shape the function of a single biological design at the point where it starts to be used, but also to sculpt the surrounding evotype landscape such that evolutionary change that can occur during its use is either managed or exploited through choices in the variation, function and selection that are actually present within the system itself. And this allows us to then carefully craft the evolutionary potential of our designs and harness evolution as a first-class citizen during the biological design process. At first, this might seem like a really challenging task and perhaps even impossible, and it did to us, I think, initially, but what you find when you start to look across the field is that there are actually many principles and tools that are going to be necessary to start shaping these factors. So, for example, variation can be reduced by avoiding, say, repetitive sequences, or we can actually enable specific jumps in sequence space by designing targeted recombination sites. Similarly, for something like selection, we can aim to reduce the burden of our designs by maybe introducing some self-regulation and kind of trying to reduce the selective pressures that would be present. Or we can actually try and couple function to growth such that evolution naturally improves the functionality of our design over time using approaches like directed evolution. So as you can see, we have some of the principles and tools, I think, to start shaping the evolutionary potential of biological designs, but we likely need more. And I think most importantly, we need a more quantitative understanding of the role of these different processes and tools to be able to develop more predictive models which will actually help accelerate their actual use. And this is something that my group is actively exploring to try and make this idea of evotype engineering an actual reality. So to end, I hope that I've shown you that engineering and evolution share many similarities, but that we may want to carefully think about how we support open-endedness in our design processes, how we handle some of the limited knowledge of biology and the kind of the big reality gaps between our models and experiments, to think about whether we actually have all the stepping stones that are needed for effective biological design, or whether there are key technologies that we're currently lacking. And finally, how we should integrate evolution into the wider bioengineering life cycle. So a lot of, I think, really important questions to think about. I don't really have answers to any of these, but I'm hoping with all the bright young minds here, there'll be some really lively discussions about how some of these aspects might be addressed. So that's me done. I wanted to thank you all for listening, and I also wanted to thank my whole lab and collaborators, as none of this work would have been possible without some of the weird and wonderful perspectives that they bring to the science that goes on in my group. And shamelessly, I'd like to plug a few opportunities. So if this sounds interesting to you, then I have a number of open PhD and postdoc positions in my group, and you can find out more information at my website, also about some of the preprints and papers on this topic and other related topics. And with that, I'll end and thank you for having me.